Hello everyone, I welcome you all for this session. A very good morning to all of you. Can you see me? Can you hear me? I guess the visibility audibility is great. Can someone give me a thumbs up in the chat box if I'm clearly visible audible? Can I start the session ahead? Okay, so today in the morning we have started a crash course of the general pathology. We have started the first chapter, that is cell adaptation, cell injury and cell death, right? So in the first lecture I have already completed cell adaptations. I guess you can see the 5 type of adaptations we have completed in the last lecture. In a comparative manner I taught you the 5 type of adaptations. After that we have already started the cell injury, the second chapter, cell injury. In the cell injury I already taught you the morphology of the cell injury, right? We have studied the morphology of the cell injury, the reversible cell injury, the irreversible cell injury. I taught you the changes in the cell in the reversible and irreversible cell injury. We have already understood it, I guess. So now it's time to start the mechanism of cell injury. So in this video, I will be first teaching you the 5 mechanisms of the cell injury. Now, whether you talk about reversible or irreversible, what are the mechanisms of the cell injury? You already know the differences between reversible and irreversible morphologically in a cell. So there are 5 mechanisms. I will be teaching you all 5 one by one in the form of the 5 flowcharts. I expect you to draw 5 flowcharts with me so that all the MCQs, as well as if it is coming in theory question in your exam, you will be able to retain it better in the form of the diagram and flowchart. Let me start with the first one, ATP depletion. What do you mean by ATP depletion? The first mechanism of the cell injury is ATP depletion. What do you mean by that? Okay, can you see here? This is the blood vessel of a cell. All cells require blood. Why? Because blood contains oxygen, the pure blood. Cell require oxygen basically. Cell don't require blood. Cell require oxygen. So cell take oxygen, and from the oxygen, the cell do oxidative phosphorylation in the mitochondria. All cells have mitochondria. So in that mitochondria, the oxidative phosphorylation takes place aerobically and ATP is formed. So mitochondria is the powerhouse of the cell in which ATP are formed. You got my point? Now imagine if there is ischemia to any cell. If any cell there is ischemia, what do you mean by that ischemia? The cell is not getting the blood. If any cell do not get the blood, the mitochondria will not receive the oxygen. So oxidative phosphorylation will not take place in the mitochondria and ATP will not form. There is less oxidative phosphorylation in the mitochondria, no oxidative phosphorylation in the mitochondria and no ATP. This is known as depletion of the ATP and it will lead to cell injury. How? Why ATP is required? Why it leads to cell injury? You got my point? Till now, you got what is there. So I would like to draw a diagram for you. So can you see a cell, a beautiful cell? Yes. Can you see the blood supply of the cell? This is the blood supply of the cell. This is the blood vessel. Now imagine there is ischemia. There is hindrance in the blood supply. Due to the ischemia, the mitochondria of the cell is not getting the oxygen. So inside the mitochondria of the cell, there is no oxidative phosphorylation. So this is the mitochondria inside which there is less oxidative phosphorylation. So less ATP formation. So inside the cell, we have ATP depletion basically. Now in this cell, there is cell injury by 3 mechanisms. Because of ATP depletion, 3 things will happen. What 3 things will happen? Now see, on the cell surface, we have a sodium potassium ATPase. Sodium potassium ATPase, right? So what does this do? Sodium potassium ATPase, what does it do? Normally from this, sodium goes out. Sodium goes out of the cell and potassium comes inside the cell. It is a transporter. It is present on the surface of the cell, and for its functioning it requires ATP. It requires ATP. So now in the cell, as I've told you, ATP is absent. So this transporter will not work. Sodium potassium ATPase will not work. So what will happen? Sodium will not go out. Sodium will remain inside the cell only. Sodium will remain inside the cell only. It will not go out. And wherever is the sodium, water, it drags water. It osmotically drags water. So sodium remains inside the cell, water will also remain inside the cell, and because of the water, the cell will swell. So this cell will swell, because of which the cell will swell. Not only swell, the cell organelles inside it, that is endoplasmic reticulum, mitochondria, lysosome, they will also swell. So all the cell organelles, that is endoplasmic reticulum, the mitochondria, everything will swell. So the cell swell, the cell organelles will swell, and because of which the cell surface will also have blebs. So this is the first mechanism by which we are having cell swelling, cell organelle endoplasmic reticulum swelling, mitochondria swelling, and we have blebs. Give me a thumbs up. That is non-functioning of sodium potassium ATPase. I will show you how to write in your exam. First understand. This is the first thing. So this is the thing written here. The diagram are from Robbins. All the diagrams are from Robbins. So can you see here? It is written. Can you see here? So the ischemia is there, no oxygen, no oxidative phosphorylation, no ATP. Since no ATP, the first thing happens is sodium potassium pump is not functioning. Since it is not functioning, there is more sodium inside the cell, there is more water inside the cell, and there is less potassium. The potassium will go out. Will go out. So there is cell swelling, endoplasmic reticulum swelling, cell swelling, and there are blebs. Give me a thumbs up. So can you see this diagram? The cell, there are blebs. On the surface of the cell, there are blebs. The cell is swelled. Can you appreciate the swelling? And can you appreciate the endoplasmic reticulum is also swelled? The mitochondria is also swelled. Give me a thumbs up. So this cell is undergoing injury. Number one mechanism, because of ATP depletion. Now coming on the second mechanism. Now again, I would like to draw the same cell. This is a cell inside which ischemia is there, and because of ischemia it is not getting oxygen, it is not doing oxidative phosphorylation, so mitochondria is not forming ATP. So ATP is not formed, this cell is having ATP depletion. Now this cell will try to form ATP by some other mechanism. Normally in mitochondria, oxidative phosphorylation takes place in presence of oxygen, ATP is formed. That is known as oxidative phosphorylation. But now this cell will try to form ATP anaerobically. Anaerobic phosphorylation will take place, and anaerobically in absence of oxygen, since there is ischemia, there is no blood supply and there is no oxygen. So this cell will try to do anaerobic phosphorylation in which ATP is formed. Some ATP is formed, a little bit ATP is formed, but the end product of anaerobic phosphorylation is lactic acid. So lactic acid is formed in the cytoplasm. Because of lactic acid formed in the cytoplasm of this cell, the pH of the cell become acidic. The pH will become acidic, and the nucleus of the cell, this is the nucleus. In pH acid, the nucleus undergo clumping. So the nucleus undergo clumping because of this mechanism. Give me a thumbs up. So you can see it is written here. In this, you can written here, you can see because of now there is no ATP now. So cell will try to form ATP anaerobically, anaerobic glycolysis. So because of which lactic acid is formed, lactic acid will cause acidic pH that will lead to clumping of the nucleus. So see this cell, the nucleus is clumped. Can you see the clumps in the nucleus? So the cell is swelled because of this. Cell have blebs because of this. All the cell organelles, endoplasmic mitochondria swelled because of this mechanism. The second mechanism, that is lactic acid formation, it will lead to clumping of the nucleus in the same cell. Give me a thumbs up. Now the third and the last mechanism, again I would like to draw the same cell. Now this is a cell inside which due to ischemia there is no oxygen. No oxygen means oxidative phosphorylation do not take place in mitochondria, and there is no ATP formation. So ATP depletion is there. Now this is the endoplasmic reticulum of this cell. On the surface of the endoplasmic reticulum, normally ribosomes are bind. You know, rough endoplasmic reticulum. These are the ribosomes on the surface of the endoplasmic reticulum, and ribosomes form protein when they are present on the surface of the endoplasmic reticulum. For binding on the surface of endoplasmic reticulum, they require ATP. But this cell don't have ATP. Since they don't have ATP, so the ribosome will disperse. They will disperse from the endoplasmic reticulum. They will go away from the endoplasmic reticulum, dispersion of the ribosome. And since ribosome got dispersed, they will not form protein. So there is no protein formation inside the cell. Now, no protein formation means no enzyme formation. All the enzymes are made up of protein. No cell membrane formation. Cell membrane is made up of protein. So ultimately, this cell will injured and it will die. So that is the third thing. Give me a thumbs up. So what is the third thing? The third thing is in front of you. Now there is no ATP. No ATP means ribosome will detach from the endoplasmic reticulum. And there is no protein synthesis, no enzyme, nothing. So you can show the all three mechanism in one diagram. In one diagram, how you will show? Draw a big cell. I will also try in front of you only. So draw a big cell like this and draw the blood supply of this cell. In the blood supply, show there is ischemia. So there is ischemia. This cell is getting less blood because of ischemia. Okay. Let me draw. This is the nucleus of the cell. This is the mitochondria of the cell. This is the endoplasmic reticulum of the cell, and these are the ribosomes on the surface of the endoplasmic reticulum. Now you tell me, and this is a pump, the sodium potassium pump, sodium potassium ATPase pump. Now you tell me what will happen. What will happen? Now because of ischemia, there is no oxygen. Okay, you draw all three mechanisms. What will happen? No oxygen. So mitochondria may oxidative phosphorylation will not take place. So ATP will not form. So ATP depletion is there. Because of ATP depletion, the first thing, this pump will not function. This pump will not function. So sodium will not go out and potassium will not come in. So sodium will remain in only and water will also remain in. And these will cause three things. What three things? They will cause cell swelling, endoplasmic reticulum swelling, mitochondria swelling, and blebs. So these are the changes because of this mechanism. So I will change the color to show you the second mechanism. The second mechanism, because of lack of oxygen, lack of ATP, the cell will try to do it anaerobically. Anaerobic glycolysis will take place, because of which lactic acid is formed. That will reduce the pH of the cytoplasm acidic. That will cause clumping of the nucleus. So this is the second mechanism. The nucleus got clumped. The nucleus got clumped. The third mechanism, again, I will change the color. Okay. Now, ribosomes attach on the surface of the endoplasmic reticulum with the help of ATP. No ATP means ribosome will detach. The ribosomes will detach. They will disperse from the surface of the ribosome, and there is no protein synthesis. No protein synthesis means no enzyme, no cell membrane, no structural changes, no structural components. So you can see the three changes with the three colors. Everyone give me a thumbs up. Draw three flow charts and draw in one cell. If you want to draw three different cells, draw one mechanism in each of them, and if you want to combine them, you can draw in one cell also. Everyone give me a thumbs up. So the diagram is from the Robbins. Everyone give me a thumbs up. You can see this is a sodium potassium pump. Normally it takes sodium out, and normally it takes potassium in, but for the functioning ATP is required. If there is no ATP, this will not happen. Sodium will remain in only, and it will hold the water. These are the ribosomes. On the surface of the ribosome, protein synthesis take place, and ribosome, till it is present on endoplasmic reticulum, this will happen, otherwise it will not happen. So draw the three flow chart. This is the first flow chart in front of you. Due to ischemia, inside the mitochondria, there is no oxidative phosphorylation. No oxidative phosphorylation means sodium pump will not function. If sodium pump will not function, the sodium and water will remain inside the cell only. They cannot exit. And this sodium and water remain inside the cell only. So cell will swell, endoplasmic reticulum will swell, and blebs will be formed. This is the first mechanism. Give me a thumbs up. The second mechanism is in front of you. Now cell try to do anaerobic glycolysis because the cell want ATP from any source. So some ATP is formed, but glycogen convert into lactic acid. Lactic acid cause acidic pH, and that will cause clumping of the nucleus. Give me a thumbs up. The second mechanism. The third mechanism you have to write like this. Due to the decreased ATP, the ribosomes will detach from rough endoplasmic reticulum. They will go away and they will not synthesize protein. No protein synthesis means no membrane formation, no cytoskeletal formation, no enzyme formation. So imagine a cell in which the nucleus is clumped, the cell is swelled, the endoplasmic reticulum is swelled, the mitochondria is swelled, and blebs are there, and the ribosomes are detached from the mitochondria, and no protein formation. So such cell is undergoing cell injury. So these are the three flowcharts from ATP depletion. Give me a thumbs up. We are done with ATP depletion. Draw three flowcharts and three diagrams. You will get full marks. Everyone give me a thumbs up. Coming on the second, mitochondrial damage. The second one is the mitochondrial damage. Here also I will give you three flowchart and three diagrams. Mitochondria is the powerhouse of the cell. We all know mitochondria may ATP formation take place. So when mitochondria is damaged, now listen now. Listen. Okay. So this is the mitochondria of a cell. Right, mitochondria have two membranes. These are the two, inner and outer membrane of a mitochondria. Inside mitochondria, oxidative phosphorylation take place, in which oxygen get converted to water, and during this process ATP is formed. This is known as oxidative phosphorylation, in which oxygen get converted to water, and during this phenomenon many ATPs are formed. This is known as oxidative phosphorylation. Now imagine this mitochondria is damaged. This mitochondria is damaged due to any mechanism. So normal oxidative phosphorylation will not take place, and abnormal oxidative phosphorylation will take place. What do you mean by that? Normal oxidative phosphorylation will not take place means ATP will not form. ATP will not form, and due to ATP depletion, you already know what three things will happen. The sodium potassium pump will not function, the ribosome will disperse, you know, and anaerobic glycolysis will take place. So you already know. So there is combination of multiple flow charts together. They all are not separate, they are integrated with each other. So that flowchart you can integrate here, right? So ATP will not form because normal oxidative phosphorylation not take place. Abnormal is taking place. So normally oxygen get converted to water, but abnormally here oxygen will get converted to reactive oxygen species, right? So here reactive oxygen species are formed, water is not formed, and these reactive oxygen species, ROS, will cause damage to the cell. They will cause damage to all the cell membrane, nucleus, everything, right? And the third thing, so normal will not take place, ATP will not form. Abnormal will take place, ROS will form, right? And the third thing, between the two membranes of the mitochondria, there is a protein which is known as cytochrome C. Cytochrome C, it never comes in the cytoplasm. When mitochondrial damage is there, it will rupture and cytochrome C will come in the cytoplasm after leaking. This cytochrome C will cause apoptosis of the cell. Whenever cytochrome C comes in the cell, it will cause apoptosis. Give me a thumbs up. So these are the three things which will happen due to mitochondrial damage. The same diagram from the Robbins is in front of you. You can see this diagram in front of you. Everyone give me a thumbs up. So normal oxidative phosphorylation not take place, so ATP will not form. Abnormal will take place, so ROS will form. ATP नहीं बनेगा और ROS बनेगा क्योंकि normal नहीं हो रहा है, abnormal हो रहा है. Give me a thumbs up. And the third thing, you can see cytochrome C is leaking out, which is causing apoptosis. So these are the three things. Write three flowchart. Mitochondrial damage, normal oxidative phosphorylation not take place, So no ATP formation, the first thing, right? And the second, abnormal oxidative phosphorylation will take place. So ROS are formed. ROS is reactive oxygen species. And the third one, it is release of cytochrome C, which will cause apoptosis. Give me a thumbs up. So I'm done with the second injury also. So these are the three things. Normal will not take place, so no ATP formation. Abnormal will take place, so ROS formation. And due to the release of cytochrome C, it's apoptosis. Everyone give me a thumbs up. That is mitochondrial damage. We are done with ATP depletion. We are done with mitochondrial damage. And now I'm directly coming on free radical injury. These you can read by yourself, not very important one for the exam. So I'm directly coming on because the time is less now. So I'm directly coming on the free radical injury. The fifth mechanism, the free radical injury. Just a second. The free radical injury, right? This mechanism. What do you mean by free radicals, by the way? What do you mean by free radicals? Okay. As I've told you, inside the cell, this is a cell. Inside the cell, there is a mitochondria, right? Just a second. Inside the cell, we have mitochondria. Okay, this is a cell. Inside the cell, we have mitochondria. In the mitochondria, oxidative phosphorylation takes place. What do you mean by oxidative phosphorylation? What happens during oxidative phosphorylation? Inside mitochondria, what happens? So basically, oxygen get converted to water. This is oxidative phosphorylation during which ATP are formed. Give me a thumbs up. But this don't take place directly. There are intermediates. There are three intermediates formed. First, oxygen get converted to superoxide ion. O2 negative, this is known as superoxide ion. Then superoxide ion get converted into hydrogen peroxide. This is known as H2O2. And finally, H2O2 get converted into hydroxyl ion OH negative. And this OH negative get converted to water. So these three intermediates are known as free radicals. There are three free radicals. One is superoxide, O2 negative. One is hydrogen peroxide, H2O2, and third is hydroxyl ion OH negative. And this is how water is formed. Give me a thumbs up. So these are the three free radicals. You can see here. So how they are formed. The same diagram is drawn here. So first oxygen get converted to superoxide O2 negative. Then superoxide get converted to hydrogen peroxide H2O2. And then H2O2 get converted to OH negative hydroxyl ion, and finally water is formed. Give me a thumbs up. This is how it takes place. So these three are not good. They cause cell injury, multiple cell injury. So this is the thing. So what is their definition? They have a single unpaired electron in their outermost orbit. They all three have single unpaired electron in their outermost orbit. That's why they damage the cell. They are highly reactive. These are the reactive oxygen species, and they are formed in the inner mitochondrial membrane, and they damage the cell. Give me a thumbs up. Everyone give me a thumbs up. So here I would like to tell you two reactions. What I have taught you here, oxygen get converted to superoxide. Superoxide get converted to H2O2. How H2O2 get converted to OH negative? Can you see here? H2O2 get converted to OH negative by two mechanisms. H2O2 get converted to OH negative by two mechanisms. If it occurs in presence of iron, it is known as Fenton's reaction. And if it occurs in presence of oxygen, it is known as Haber-Weiss. You got my point? So again, I am writing. For those who don't understand this, I am writing it again. So first oxygen is there. It get converted to superoxide, O2 negative. Then it converted to H2O2. You have to learn the sequence. H2O2, right? Hydrogen peroxide. Then it get converted to OH negative. And finally, water is formed. Give me a thumbs up. Now I'm talking about this reaction. H2O2 to OH negative. If it takes place in presence of iron, it is known as Fenton reaction. And if it takes place in presence of oxygen, it is known as Haber-Weiss reaction. Haber-Weiss. So these are 2 mark questions, and you should be able to answer it. Everyone give me a thumbs up. So that is the free radical injury. That is the two type of reactions here. So I have already taught you there are three type of free radicals: superoxide ion, hydrogen peroxide ion, and hydroxyl ion. Write their names, write their formula. So that is three type of free radicals, right? What they cause? They damage protein, lipid, nucleic acid, carbohydrate. Everything is damaged in the cell because of them. So they basically damage the cell. But they are formed in all cells. Now I taught you whenever in any cell, this is a cell. Inside the mitochondria, oxidative phosphorylation take place. So during the oxygen to water conversion, these are the three intermediates. These are always formed in between. Whenever oxygen get converted to water, these three are formed. First superoxide is formed, then H2O2 is formed, and then OH negative, and then, so these are the byproducts. So for their removal, God has given three enzymes in our cell. In all of our human body cell, these are formed as a byproduct, and these are the byproducts, right? And God has given three enzymes to remove them. What are the name of the three enzymes? The name of the three enzymes are in front of you. SOD, it is superoxide dismutase, glutathione peroxidase, and catalase, right? So this is the summary in front of you. So what does each enzyme will do? Superoxide mutase. Okay. Can you see the conversion, or you want me to write it again? Okay, I will write it for you. See, what is the sequence? Oxygen get converted to O2 negative. O2 negative get converted to H2O2. H2O2 get converted to OH negative, and OH negative get converted to water. You can see what are the free radicals here. You can see 3 free radicals here. This is the first one, O2 negative, second one H2O2, third one. So this and this. These are the 3 free radicals, right? Now let me tell you the 3 enzymes. So the first enzyme, superoxide dismutase, convert O2 negative to H2O2. The second enzyme, glutathione, convert H2O2 to OH negative. And the third enzyme, catalase, convert H2O2 directly to water. So these are the 3 enzymes present in front of you, catalase. In simplicity, you can learn like this and write the function of each enzyme in your exam. So basically, all these enzymes are for the removal of the free radical. Give me a thumbs up. What are the 3 enzymes? Superoxide dismutase, what it is converting? Glutathione, what it is converting? And catalase, what it is converting? Everyone give me a thumbs up. So all these 3 enzymes basically pushing towards the water. Ultimately, we want them to get converted into water because they are reactive. Water is non-reactive. We want to convert the reactive oxygen species into water, and these enzymes are helping in that. So what are the 3 enzymes you can see in front of you? So superoxide dismutase convert O2 negative to H2O2. Second, glutathione converts H2O2 to OH negative, and catalase convert H2O2 directly to water. Give me a thumbs up. So these are the superoxide dismutase. As I have told you, what does it do? It converts O2 negative to H2O2, right? So you can see this is the function. Superoxide dismutase, what it is doing, right? Second, glutathione, it is present in mitochondria and cytoplasm. What it is doing? Glutathione convert H2O2 to OH negative, right? That is the second. And catalase is present in peroxisomes. It directly convert H2O2 to the 2 waters. That is the thing. Everyone give me a thumbs up. So this is the summary. This is the summary of free radical. How many types of free radical are there? 3. So what are their names? O2 negative, H2O2, and OH negative. How they are formed? Write the reactions, and what are the 3 enzymes? SODs, that is superoxide dismutase, glutathione, peroxidase, and catalase. So write the 3 free radicals and write the 3 enzymes which are required for their removal. Everyone give me a thumbs up. So which free radical are formed in which step, and which enzyme convert which of them into this, and what is Fenton's reaction and what is Haber-Weiss reaction. So you have to write everything about that in the free radical injury. I am done with cell adaptations. I am done with cell injury. Now it's time to study cell death. Cell death is of 2 types. What are the 2 types of cell death? Apoptosis and necrosis. So now I'm going to teach you 2 ultra important topic in this lecture: apoptosis and necrosis. Expect 1 question in your university exam from this. It is a fixed every year, either apoptosis or necrosis, one of them come in every year university exam in paper 1 of pathology. So you can say it is a paper leak or whatever you say, but it will always come in your exam. So one of the question is fixed from apoptosis and necrosis. It is that way important. So you cannot afford missing it, right? You want to secure 1 question 100% for your university exam. Apoptosis necrosis में से एक question पक्का है. Either complete 5 mark question from each of them or some 3 mark question from their subheadings. Everyone give me a thumbs up. Everyone. Shall I start? Apoptosis necrosis. I will start with apoptosis. The 2 types of the cell death. So apoptosis. What is apoptosis? What are the 2 types of cell death? Why cell is given a choice? The 2 types of cell death. The 2 types of cell death, apoptosis, necrosis. Is there any difference in them? Yes. Apoptosis, if you want me to say in 1 line, in 1 word, it is cell suicide. So I will discuss apoptosis in these headings. First, I will tell you the definition, then types, then mechanism, then morphology, diagnosis, differences from necrosis. In your exam also, if it is coming for 5 marks, I'm expecting you to write in these headings only. Don't write paragraphs. Divide the apoptosis into following headings. Start with definition. In 1 word, if you ask me to say the definition, it is the suicide of the cell. The cell is committing the suicide. Now, why the cell is committing the suicide? No one likes to do the suicide. Now, if someone is doing suicide, there may be a reason behind it. So, why the cell is committing suicide? Cell may also have a reason to commit suicide. इच्छा से तो कोई नहीं करता ना सुसाइड कर रहा है तो कोई मजबूरी होगी तो सेल का क्या प्रॉब्लम है व्हाट इज द प्रॉब्लम टू दिस सेल व्हाई द सेल इज कमिटिंग दिस सुसाइड दैट इज नोन एज apoptosis. Cell may have one of the 2 problem. Either the cell feels the job is done in the body, this cell is no more required in the body. The cell will get the signal from the human body that you are no more required, your job is done, your function is done, now you should undergo suicide, right? That is the first reason. The second reason, the cell, this is a cell, this is the nucleus of the cell, and this is the DNA. Sometimes DNA of the cell is damaged too much. First, the DNA is damaged, so we try to repair them with the help of P53. But sometimes damage is too much that even P53 cannot repair it. So if the DNA is damaged beyond repair, then we ask the cell to commit suicide, right? So human body give the signal that your nucleus is damaged. Now it is beyond repair. We cannot repair it. So you should commit suicide. So cell do suicide because of two reasons. Either it is no more required in the body, its function is done, its job is done, or its nucleus, its DNA is damaged beyond repair. Give me a thumbs up. So that is cell suicide. Give me a thumbs up in short. So how the cell commits suicide? Now this is the definition of actual definition. This is just for understanding. You can write cell suicide, but this is not the actual definition. Now, for the sake of understanding, it's good. What is the actual definition of apoptosis? This is the actual definition. It is looking a difficult one from the Robbins. Let me simplify it. Always to simplify, break the language into multiple points. Now see, this is a cell. This cell have two problems. Either its job is done, it is no more required in the body, or its DNA is damaged beyond repair. Now it has received the signal that it has to commit suicide. Now signal can come from two parts. Either signal come externally. External signal will come in the form of the ligand, and the cell will get the signal that I must commit suicide. The body is saying I must commit suicide. That is external signal. Or signal will come from inside, the mitochondria. Mitochondria will give the signal to the nucleus that you must commit suicide. You are damaged now and you must commit suicide, right? So signal is either coming from outside or inside. So two type of signals are there, extrinsic and intrinsic. Anyways, whenever the cell will receive the signal, inside the cell there are many enzymes. These are known as caspase. These are normally all cells have caspase, but they are inactive. On receiving signal, the caspase will convert active. There are many type of caspase, 1, 2, 3, 4, 5, 6, many. All of them will become active. Ultimately, after becoming active, what they do? The caspase is an enzyme which degrades three portions of the cell. It degrades the cell membrane, it degrades the cytoplasm, and it degrades the nucleus and convert the cell into multiple small bodies known as apoptotic bodies. Listen now, in each apoptotic body there is a small portion of the cell membrane of the cell. In each apoptotic body there is a small portion of cytoplasm of the cell, and in each apoptotic body there is a small portion of the nucleus. So all apoptotic bodies contain small cell membrane, small cytoplasm, small nucleus, and there is no leakage outside. The entire cell is converted into multiple small bodies. That is known as apoptotic bodies. This is the job of caspase. Give me a thumbs up. Since there is no leakage, cytoplasm do not leak out. There is no bursting of the cell. There is no leakage. That cell chupchap se get converted into multiple small bodies. Give me a thumbs up. Give me a thumbs up. There is no leakage. You got my point? In the end, a phagocyte will come. The green color cell is the phagocyte. It will come and engulf all the apoptotic bodies. So this cell is disappeared from the world. This cell is disappeared from the body. It has committed suicide. So whenever a cell want to commit the suicide, whenever the cell receives signal, either extrinsic or intrinsic signal, cell activate caspase. After activating caspase, the cell convert into multiple small apoptotic bodies. In the end, a phagocyte come and engulf all the apoptotic bodies. During this entire process, there is no leakage of cytoplasm outside. Bahar leakage nahi hoga, so there is no inflammation. Neutrophil will not come to know that something is happening. Chupchaps, it is a silent process. If someone is committing suicide, he don't take a mic speaker and announce everywhere that I am committing suicide. He do it silently now. In the same way, the cell is also doing it silently. Nobody will know that cell is committing suicide. The cell just disappears. It is like that. You can understand that. So there is no leakage. There is no inflammation. कुछ हल्ला गुल्ला नहीं मचेगा, कोई inflammation नहीं होगा, no neutrophil will come. There is no leakage, there is no inflammation. Cell membrane will remain intact till end. You cannot say that during the conversion of cell into multiple apoptotic bodies, there is hole in the cell membrane. No, there is no hole. There is no gap. It remains intact. Cell membrane is intact. That's why there is no leakage, no inflammation. These are the differences between apoptosis and necrosis. Necrosis is the second type of cell death in which cell burst. वो चिल्ला चिल्ला के मरेगा कि मैं मर रहा हूँ. You got my point? ये silently suicide करेगा. वो चिल्ला चिल्ला के बताएगा कि मुझे ये लोग मार रहे हैं, मुझे बचाओ. वैसा. So that is not suicide. That is murder. So you can say necrosis is murder and apoptosis is suicide. Both are death, but the mechanism is different. One is suicide, one is murder. So in suicide, patient does it silently. The person does it silently. And murder, the person shouts, please save me, I'm dying. But still it dies. So that is the two type of cell death: apoptosis, necrosis, the suicide and the murder. So here it is intact till end. So no leakage, no inflammation. In contrast to necrosis, the cell burst. वो चिल्लाएगा, right? The cell burst. सबको बुलाएगा कि मुझे बचाओ. So there cell membrane is defective. There are holes in the cell membrane. So there is leakage, there is inflammation in necrosis, not in apoptosis. Currently, I am teaching you apoptosis. Give me a thumbs up. Everyone give me a thumbs up. So what is the definition? This is the definition. So it is a pathway of cell death. Apoptosis is a pathway of one of the pathway of cell death in which which is tightly regulated program in which the cell which is destined to die, the cell which is destined means the cell which gets signal for dying, the cell which is destined to die activate enzyme known as caspase inside them. The cell activate caspase inside them. Caspase degrade the cell into multiple apoptotic bodies. The cell is phagocyzed. In the end, a phagocyte will come and the phagocyte will engulf the cell. There is no leakage, there is no inflammation. So write down 2 negative findings compulsorily in the definition in your exam. These are 2 negative. You will say, ma'am, agar nahi hai to kyun likhna hai? Why to write in your exam if there is negative? The negative findings are important because they are differentiating it from the second type of cell death, necrosis. So say the definition. Apoptosis is a type of cell death in which the cell which is destined to die activate caspase inside them, the enzyme caspase. Caspase degrade the cell into multiple apoptotic bodies. In the end, a phagocyte will come and engulf the apoptotic bodies. There is no leakage, the cell membrane is intact, there is no inflammation. This is the definition of apoptosis that you have to write in your exam. Give me a thumbs up. So after definition, after diagram, it is a way of eliminating unwanted cell from the body. Can I say yes? It is. Now, apoptosis takes place of a single cell and necrosis takes place of a group of cells, right? Apoptosis is always a single cell and necrosis takes place of a group of cells. Again, a difference, right? So it is programmed cell death. It is genetically programmed and it requires ATP. For apoptosis, ATP is required. It is energy dependent. यहाँ पे मरना भी मुफ्त में नहीं है. For if the cell want to die, cell have to invest ATP. Otherwise, यहाँ पे free में cell मर भी नहीं सकता, right? So it is ATP dependent. And necrosis one is free. It is passive. So this one is active and the necrosis is passive. Again, a difference between apoptosis and necrosis. You got my point? We are done with definition. Let's come on the type of apoptosis. Apoptosis is of 2 types, physiological and pathological. So can you give me few examples of physiological apoptosis? Few example of physiological apoptosis body में कहाँ होता है? Pathological तो occurs in many diseases. Physiological का कोई example दोगे? Physiological apoptosis का few examples. Okay. The best example is embryogenesis. What is embryogenesis? Okay. Can you see my hand? Can you see my fingers? The fingers, the digits. So this is the hand. This is the hand. So whenever we were embryo, whenever we were embryo, the hand is not like this. Inside the uterus of the mother, the hands and the feet are like this, Now what happened? To convert it into digits, the cells here, and here, they die. So that the multiple fingers and digits, it converted into multiple fingers and digits. So the cells, the red portion is the cells, they are dying. They are dying because they are no more required. You got my point? So during embryogenesis, not during the formation of the digits, but at many places in the body organs, apoptosis take place for giving the shape to the body, for giving the shape to the embryo, to the fetus. Give me a thumbs up. Everyone give me a thumbs up. That is embryogenesis. One of the best example is formation of digits. This is how digits. Initially, we all have like a block. The hands and the feet were like a block whenever we were fetus inside the uterus of the mother. But during embryogenesis, the cells between the digits, they die so that the digits separated. So we have digits now. Give me a thumbs up if you got my point. So this is the example, the best example you can quote for physiological. Otherwise, we have WBCs inside our blood, right? One of the WBC is lymphocyte. Ideally, lymphocyte should kill foreign. It should not kill the self. That is the function of WBC, no? They should kill foreign. Sometimes lymphocytes become mad. They kill self. These are known as self-reacting lymphocytes. So every day in our body, some of the lymphocytes convert mad. And they are self-reactive lymphocytes. So body eliminate them. Body asks them to do the suicide. You are mad and you just commit the suicide. You are killing the self. How you can kill the self? You are supposed to kill the foreign. If you can't kill foreign, if you are killing the self, you commit suicide. So self-reactive lymphocytes. It is a physiological process. Give me a thumbs up. So these are the examples of physiological. There are many diseases in which pathological apoptosis takes place. So what are the example of pathological apoptosis? In AIDS, you know HIV, AIDS. AIDS means there is a virus, HIV virus. HIV virus selectively could do the apoptosis of CD4 T lymphocyte. No other cell will die. Only CD4 T lymphocyte will die. Everyone give me a thumbs up. So in hepatitis virus, they selectively cause apoptosis of the hepatocytes of the liver. So these are the examples. Give me a thumbs up. So we have done the definition. We have done the types, physiological, pathological, few examples here, few examples here. Give me a thumbs up. The most important, difficult thing to understand in apoptosis is the mechanism. Now see, I'm going to teach you the mechanism of apoptosis in a super duper simplified diagrams. I have drawn these diagrams for you. It is nowhere given in no book, not in Robbins, not in Hirschman, not in any book. I have drawn these simplified diagram to explain you. You just have a look on that. Then in the end, you give me a feedback that how you get the concept here. So let me start the mechanism of apoptosis now. As I told you, in apoptosis, this is a cell. This cell is committed destined to commit suicide now. This cell is committed for the apoptosis. Apoptosis is cell suicide. So it will receive signal from two sources. Either the signal will come from outside, or the signal will come from inside. So it is extrinsic pathway, it is intrinsic pathway. In extrinsic pathway, the signal will come from outside. In intrinsic pathway, the signal will come from inside. So there are two mechanisms in apoptosis. What are the two mechanisms? Extrinsic pathway, intrinsic pathway, depending signal kahan se aa raha hai. Give me a thumbs up. In each pathway, listen now, in each pathway, extrinsic pathway as well as intrinsic pathway, there are two phases. Initiation, execution, initiation, execution. First, give me a thumbs up on this. Everyone, jitne audience ho, utne sabke sab thumbs up do. So there are two pathways in apoptosis, extrinsic and intrinsic, depending signal is coming from where. If signal is coming outside the cell, it is extrinsic. If signal is coming from inside the cell, it is intrinsic. In each of them, whether it is extrinsic or intrinsic, there are two phase. So initiation, extrinsic, execution, extrinsic. Initiation, intrinsic, execution, intrinsic. So this is the thing. Till now you got it. Now listen the next thing. So first I will teach you initiation of extrinsic, then I will teach you initiation. I will not complete execution. Then I will teach you initiation of intrinsic. First I will teach you initiation of both of them, because execution of both of them is common. So I will teach you in common. So now I am going to teach you three movie stories. The first story will be initiation of extrinsic. That is the first half. After intermission, I will leave it. I will not tell you the second part. I will tell you later on. Then another story I will start. Initiation of intrinsic. Again, I will leave it at intermission. After intermission, the story is common. We will combine the two stories, movie stories, and we will combine in a common convergence. So execution of both of them is common. You got my point? So let me start with initiation of extrinsic. Extrinsic, the signal will come from outside. Fatafat thumbs up dete jao har point pe. So let me start with initiation of extrinsic, the signal will come from outside, right? Starting with the story. So I am starting extrinsic pathway ka initiation phase, the first phase, right? So here signal come from outside, as I have told you. See, this is a cell. All cells have death receptor on its surface. All cells, why my remote is not working? I guess the cells are not working. Give me a minute. Okay, I will try now. Yeah. So can you see this is a cell? On the surface of the cell, we have death receptors. Can you see these are the red colored death receptors? On all cell surface, we have these death receptors. These are known as death receptor. Death receptor are of two types. The first death receptor is CD95, also known as FAS. FAS or CD95 receptor, one and the same thing. ये एक दूसरे के synonym है. FAS and CD95. The second is TNF. It is not shown here. TNF. TNF is the second one. So one of the death receptor is present. Can you see this is a death receptor? This is a death receptor. They are present at a distance from each other. They are present at a distance from each other. Now, this is the signal coming from outside in the form of the ligand. This is the signal. It is coming from, you can see the signal is coming from outside in the form of the ligand. Now the body is sending the signal to this cell. You please commit suicide. You are no more required in the world. You are no more required in the body. Your job is done. Your function is done. Now you please commit suicide. This is the signal. The signal is in the form of the ligand. Now this ligand will come and bind with the death receptor. What is the name of the death receptor? It is CD95 FAS receptor. Give me a thumbs up. It is CD95 FAS receptor. Give me a thumbs up. Give me a thumbs up. So can you see this? Now let me zoom this portion. I'm drawing this portion in a zoom version for you to explain you, right? So can you see the zoom version? So this is the ligand coming here and binding with the receptor. The ligand is coming and binding. So FAS ligand coming and binding with the FAS receptor. CD95 ligand coming and binding with the CD95 receptor. Once it bind and give the signal here, so multiple after this, can you see the combination? Can you see the binding? Appreciate the binding. So after that, multiple death receptors come closer. So these multiple death receptors come closer. Can you appreciate? यहाँ पे ये दूर थे. There was some distance between them. And now once the ligand come and give the signal here, multiple death receptors come together and they form a domain. The name of the domain is FAD, F-A-D-D. It is FAS associated death domain. It is written by this name in Robbins only. It is FAS associated death domain, FADD. Give me a thumbs up. So because of the multiple death receptors are coming together, because ligand is coming and giving the signal, a domain is formed. FAS associated death domain. Now this is the interior of the cell. In the interior of the cell, these are the caspases. Multiple caspases are present, but all of them are inactive. As I have told you, caspase number 1, 2, 3, 4, 5, 6, 7, 8, 9. All of them are inactive. Because of the formation of FADD, the first caspase which become active is caspase 8. So pro-caspase 8 converts into caspase 8. Everyone give me a thumbs up. You got the story? So there is a cell. The cell contains death receptor. The name of the death receptor is CD95 or FAS, right? Now a ligand is coming. The ligand is the external. So the signal, it is carrying the signal from outside. It is binding with the death receptor and giving the signal to the death receptor. Now you have to commit suicide. Body, you do not want you more in the body. You just commit suicide. So on receiving the signal, multiple death receptors come together and they will form a domain. The name of the domain is FAS associated death domain, because of which procaspase 8 convert into active caspase 8. So caspase 8 is the first caspase which get activated in extrinsic pathway. Currently I am teaching you extrinsic pathway. This is intermission. The next half of the story will be tell you later on. So intermission कहाँ छोड़ रही हूँ मैं? याद रखना. I will start the story from here only. So I am leaving. So can you see the same diagram from the Robbins? So can you see the ligand, fast ligand coming and binding? This is the death receptor. It is binding. So multiple death receptors are coming together. It is forming a domain. The name of the domain is in front of you. It is FADD. So it is fast associated death domain, and because of which it is converting procaspase 8 into active caspase 8. Give me a thumbs up. Give me a thumbs up. So this is the story. You can write down like this in your exam. So FAS ligand is coming, binding with the FAS receptor. Multiple FAS receptors come together. They form a domain. The name of the domain is FADD, because of which procaspase 8 convert into active caspase 8. So give me a thumbs up. Give me a thumbs up. Instead of FAS, it can be TNF. So same can be, I can show with TNF. So TNF receptor will come. TNF ligand will come and bind with the TNF receptor. So multiple TNF will come closer. Multiple TNF will come closer and they will form a domain. The name of the domain here is TRAD, TNF receptor associated death domain. Ultimate result is same. Procaspase 8 convert into active caspase 8. So you can say whether it is FAS or whether it is TRAD, ultimate result is procaspase 8 to active caspase 8. So here I am ending the story of initiation phase of extrinsic. Where I am ending? I am ending it here. I will continue the story here, but later on. Now let me teach you one more story here. I will teach you initiation of intrinsic pathway, then execution दोनों का common है. Give me a thumbs up. So let me start the story now, a new story. You may have a query, you may have an anxiety, you may have a, what happens ahead ma'am, आगे क्या? So caspase 8 की activation के बाद की story क्या? I will continue it here later on, a little bit later on, after 3 minutes. So in the next 3 minutes, let me teach you initiation phase of intrinsic pathway. Give me a thumbs up. Shall I start initiation of intrinsic? So here intrinsic, the signal will come from inside, right? So I am starting initiation phase of intrinsic pathway. Everyone give me a thumbs up. Everyone give me a thumbs up. What is the stimulus here? So the signal, stimulus means signal. Signal will come from inside. It is DNA damage. Can you see a beautiful cell in front of you? Yes. You all can see a cell. You can see the nucleus. You can see the DNA. You can see the mitochondria. You can see the two layers, two membranes of the mitochondria. Between two membranes of the mitochondria, there is cytochrome C. There is a protein known as cytochrome C. It is always present here. It never comes out in the cytoplasm. Once this comes out in the cytoplasm, it will lead to apoptosis, right? See now. Now, till now, the DNA is normal. It is not damaged, right? As I told you, this is cytochrome C. It never comes out. Now, there is a mitochondria may have a gate now. Gate matlab door, darwaza. So this is known as mitochondrial transit. Why this cytochrome C not coming out of the transit? Who is preventing? Who is closing the door? No, you cannot come out. You just go inside. Who is doing that? There are two proteins. You can learn them. These are the two guards. ये तो चौकीदार हैं, दो guards हैं, दो watchmen हैं. These are the two guards. ये डंडा लेके खड़े हैं, and they are preventing cytochrome C to come out from the transit of the mitochondria. This is mitochondrial transit. Learn it like a story. This is mitochondrial transit, and there are two guards. The name of the two guards is BCL2, BCLX. Actually, these are two proteins. Learn one more, MCL. So there are three guards, BCL2, BCLX and MCL. They are closing the door. They are closing the transit and inhibiting cytochrome C to come out. That's why they inhibit apoptosis. These are known as anti-apoptotic proteins. These three are known as anti-apoptotic proteins. They close the door. They inhibit cytochrome C to come out. Normally inside me, you, everyone, this is the case, right? But whenever in any of our cell, DNA is damaged, it is damaged beyond repair. It cannot be repaired. The DNA will give signal to the mitochondria ki bhai, you just commit suicide. The cell is gone. I am gone. I am damaged and I cannot be repaired. P53 ने अपने हाथ ऊपर कर लिए. P53 cannot repair me, and you just start the process. I'm giving you internal signal. You commit suicide. So the signal is internal. That's why it is intrinsic pathway. Give me a thumbs up. Give me a thumbs up. So this mitochondria, what it will do on receiving the signal, it is internal signal. That's why intrinsic pathway. The three guards, that is BCL2, BCLX and MCL, they will be replaced by three another guards, right? So the name of another guards is Bax, Bad, Bim. You can add more. Bad, Bax, Bad, Bim, Bad. Ye, what they will do? They will open the door. They will open the door and they will allow the cytochrome C to come in the cytoplasm. So they are initiating apoptosis. That's why these are known as pro-apoptotic proteins. Give me a thumbs up. So basically anti-apoptotics are replaced by pro-apoptotics on receiving the signal. So pathologically say like this in a beautiful language. So on receiving the signal from the nucleus, mitochondria transit pay the anti-apoptotic proteins, that is BCL2, BCLX and MCL. They are replaced by pro-apoptotic proteins, that is BARX, BAD, BIM and BAD. What they will do? They will open the door. They will open the door. See, they have opened the door. Once they have opened the door, the cytochrome C is leaked out. Cytochrome C is leaked out. Give me a thumbs up. So cytochrome C bahar aake kya karega? How it will do the apoptosis? Listen like a story. In the cytoplasm, various caspase are present. Caspase 1, 2, 3, 4. Everyone is present, but all of them are inactive. What about caspase 9? This is a caspase known as caspase 9. Caspase 9 ka best friend hai APOF1. It is a protein. The full form of APOF1 is apoptotic activating protein. Right? APOF1. These two are best friends. They are never separated. And it is a rule. Till pro-caspase 9 is in combination with APOF1, it will remain inactive only. So in दोनों का pair जब तक है ये inactive ही रहेगा. Till pro-caspase 9 is in combination with APOF1, his best friend, it cannot be activated. It cannot be activated. So cytochrome C on coming out, what it will do? It will take APOF1 with him. कि तू उसको छोड़, तू मुझसे दोस्ती कर ले, I am better than him. You just leave him. So the cytochrome C will take APOF1 with him. So the pro-caspase 9 will be alone, and after that it will convert into active form. So this is how cytochrome C convert pro-caspase 9 into active caspase 9. So this pro-caspase 9 is converted into active caspase 9. So caspase 9 is the first caspase of intrinsic pathway. If you remember, caspase 8 was the first caspase in extrinsic. Give me a thumbs up. Give me a thumbs up, everyone. Jitne audience utni thumbs up. समझ में आया क्या? So the same story in the robins. The same story in the robins. You can see here. Okay, you can see these are the two membranes of, this is inner mitochondrial membrane, this is outer mitochondrial membrane, and this is cytochrome C between the two membrane. You can see this is the transit. This is the transit. On the transit, the first guard BCL2, BCLX, MCL, they close the door. But on receiving signal from the mitochondria, these are replaced by BARX, BAD, BIM and BARC. Right? They will open the transit. These are anti-apoptotic, these are pro-apoptotic. They will open and cytochrome C is coming out. On coming in the cytoplasm, the two best friends, pro-caspase 9 and APOF1. Cytochrome C take APOF1 with him. So pro-caspase 9 is alone and it get converted into active caspase 9. And it will lead to apoptosis later on in execution phase, I will tell you. So this is how you have to write in your exam. Give me a thumbs up. So on receiving stimuli, that is DNA damage. The stimulus is DNA damage, right? Beyond repair. Beyond repair is the word. So the anti-apoptotic proteins are replaced by pro-apoptotic proteins. Anti-apoptotic are BCL2, BCLX, MCL. Pro-apoptotic are BARX, BAD, BIM, BAD. These are replaced by this. So they increase the mitochondrial permeability. They will open the transit. Cytochrome C will come out and it will take APOF1 with him. So pro-caspase 9 convert into active caspase 9. Always draw flowcharts and diagrams in your exam, university exam. Never write paragraphs. Give me a thumbs up. Give me a thumbs up. So anti-apoptotic proteins are these one, BCL2, BCLX, MCL1, flip, and pro-apoptotic proteins are BARX, BAD, BIM, BARC. Give me a thumbs up. So this is the thing. And mitochondria is main. So I'm leaving the story. This is intermission. Leaving the story here. So I left this story at activation of caspase 8. I left this story at activation of caspase 9. Here, how activation of caspase 8 takes place because of death receptor, FAS, CD95, right? And FADD, fast associated death domain is formed. And here how it is activated? Because of caspase cytochrome C coming out of the mitochondria. So here signal was from outside in the form of the ligand, and here signal was from inside from damaged DNA. Give me a thumbs up. So here first caspase is caspase 8, draw flowchart, draw diagram. Here first caspase is caspase 9, draw flowchart, draw diagram. Give me a thumbs up. What ahead? इसके आगे की story क्या? Let me continue execution of both of them together, which is convergence. Give me a thumbs up. Everyone give me a thumbs up. So let me continue execution phase of both intrinsic pathway as well as extrinsic pathway. So it is a convergence point of the two pathway. Give me a thumbs up. So you can see in extrinsic pathway, it is the first caspase, caspase 8 is activated. How it is activated, I have already taught you. Right? Yes or no? Draw that flowchart. Yes? In intrinsic pathway, it is the caspase 9 is the first caspase which is already activated. How? I have already taught you. Draw this flowchart. Now it is the convergence of the both. So the caspase 8 here and caspase 9 here activate another two caspase. The another two caspase are caspase 3 and 7. So caspase 3 and 7 comes next in the role, and they activate all other caspase. So ultimately all the caspase 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 jitne bhi hote hain, they all get activated. Give me a thumbs up. And ultimately on activating, all the caspase are activated. All. If it is intrinsic pathway, the caspase 9 is the first caspase to be activated. If it is extrinsic, caspase 8 is the first caspase to be activated. Here extrinsic and intrinsic. And ultimately 3 and 7 are activated, and ultimately all are activated. So all caspase convert the entire cell into multiple small apoptotic bodies, and in the end a phagocyte will come and engulf it. Give me a thumbs up. I am done. So you can see in the end a phagocyte is coming and the cell is degenerated and phagocyte will engulf it. So this is the execution. This is the execution. I am done. So if intrinsic pathway is asked, write these two. If extrinsic is asked, write these two. And if overall is asked, write everything. Write initiation of extrinsic, write initiation of intrinsic, and then draw execution in common. Everyone give me a thumbs up. So last is execution. Mein last step kya hai? The last step is phagocytosis. Everyone give me a thumbs up. In the end, a phagocyte is coming and engulfing all the apoptotic bodies. So my question to all the students, can you see a phagocyte here in the phagocytosis? Can you see in this diagram of phagocyte? Can you see the green color cell is phagocyte? Everyone give me a thumbs up. So everyone, my question to all of you, phagocyte is a cell. It is not a human, right? Phagocyte don't have eyes. So how phagocyte recognize that it is a normal cell and I don't have to engulf this cell? But this is an apoptotic cell and I have to engulf this cell. How phagocyte recognize that it is a normal cell, I don't have to engulf it, and this is an apoptotic cell, I have to engulf it? How phagocyte recognize? Do anyone know? Pratyaksha, Kalma, Shweta, Tejaswini, anyone? Anyone knows it? Yes? So there is tagging. What is that tagging? I want to know that tagging on the apoptotic cell. What is doing the tagging? Which is attracting the phagocyte towards it. What is that tagging which is present on apoptotic cell but not on normal cell? Okay. To understand phagocytosis, understand this diagram. This is a normal cell. Can you see? This is the normal cell. This is the cell membrane of the normal cell. On the cell membrane of the normal cell, on inner side, on the inner, the word is inner. Inner side, there is a protein. The name of the protein is phosphatidylserine, PS. It is known as phosphatidylserine, PS, phosphatidylserine. It is present on the inner side. But when it converts into apoptotic bodies, during apoptosis with the help of caspase, the caspase got activated here now. The caspase 9, caspase 8, all caspase, they are converting it into apoptotic bodies. So during the conversion of apoptotic bodies, there is flip-flap, ulta-pulti, flip-flap, you know, that is externalization of the phosphatidylserine taking place. So here the phosphatidylserine is present on the external surface in apoptotic cell. So this phosphatidylserine is doing the tagging. Now phagocyte will come. So phagocyte eat only that cell which is having phosphatidylserine on the outer surface. So who is doing the tagging? The tagging is done by the phosphatidylserine. So phagocyte engulfs those cells who is having phosphatidylserine on the external surface. Phagocyte will not engulf those cells who is having phosphatidylserine on the inner surface. This is phagocytosis. Phagocytosis come for 2 or 3 marks in your exam. You have to write like this. So phosphatidylserine is a protein. It is a phospholipid, not protein, sorry. It is present on the inner side of the cell membrane normally. But during apoptosis, it is flipped to the outer surface. Flip-flap, ulta-pulti ho jayegi. This is known as externalization of the phosphatidylserine, which is causing the tagging. So a phagocyte will come and engulf only that cell which is having phosphatidylserine on the outer surface. Give me a thumbs up. Everyone give me a thumbs up. You got it? So this is how the mechanism. So the same diagram from the Robbins. You can see this is normal cell. Appreciate phosphatidylserine on the inner surface. And during apoptosis, appreciate during formation of apoptotic bodies, it is phosphatidylserine on the outer surface. So I am done. So ultimately phagocytosis occurs, but no inflammation, no leakage. So I am done with the mechanism of apoptosis. So we are done with definition. We are done with 2 types, physiological, pathological examples of each of them. We are done with the 2 mechanisms, extrinsic pathway, intrinsic pathway. In extrinsic pathway, signal come from outside. Intrinsic pathway, signal come from inside. Each having 2 phase, initiation, execution, initiation, execution. In execution, the last thing is phagocytosis. We are done with phagocytosis also. This is the summary till now. Give me a thumbs up. Yes, shall I proceed? The next heading is the morphological changes in apoptosis. Now, please, all the students, the second prof students, I'm talking. So they should be very cautious in your exam. Read your question. The examiner is asking mechanism, then draw the mechanism. Intrinsic, extrinsic, initiation, execution, all the flowcharts I taught you. The examiner is asking morphology, don't write anything about the mechanism. Morphology is absolutely different. You students even don't understand the question. कुछ भी पूछा है apoptosis है तो बस जो आता है वो लिख दो. ऐसा नहीं है. What the exact question is, you have to read the question. If mechanism is asked, write mechanism. If morphology is asked, write morphology. If diagnosis is asked, write diagnosis. Whatever asked in the question, marks will be given for that if you write that. If you write what you know, we don't give marks. If you write what is asked, then we will give you the marks, right? So this is a rule, right? So morphology is something different. How you recognize a apoptotic cell. Okay, so there are 7 morphological changes which takes place in a sequence. I will tell you all in one diagram, right? One by one. So the first change, let's start with the first one. Cell shrinkage. The first change is the cell shrinkage. Cell shrinkage, it is the earliest change. See the diagram. Can you see this is the normal cell? Now normal cell undergoes cell shrinkage. So in apoptosis, there is cell shrinkage, while in necrosis, there is cell swelling. That is the difference between apoptosis and necrosis. So can you see normal cell undergoes cell shrinkage during apoptosis, the first change. Give me a thumbs up. The second, I will tell you 7, huh? The second one, can you see in this diagram? This cell is smaller than other cells. This cell is smaller than other cells. This cell is smaller than other cells. So cell shrinkage is the first thing, right? Now, because the cell shrinkage is there, so all the organelles present here, ribosome, mitochondria, you know, endoplasmic reticulum, they all come closer. They all come closer. They all come closer. They become compact. So because the organelles become compact, the cytoplasm become more pink in color. And pink in pathology is known as eosinophilia. So the second change is increased eosinophilia because of compactness of the organelles. So all the organelles, they are tightly packed because the cell shrinkage is there. So cytoplasm become pink in color, which is known as eosinophilia. Give me a thumbs up. So the second change is eosinophilia. First is cell shrinkage, write down cell shrinkage, and second is eosinophilia. Both are shown in one diagram. Give me a thumbs up. Coming on the third change. So you can see this change, this cell is more pink than others. Can you appreciate this cell is more pink and smaller than others? This cell is more pink and smaller than others. So pink also, shrinkage also. 2 changes are done. Coming on the, can you see this cell? It is more pink and smaller than the others. Give me a thumbs up. The third change, see the nucleus. In the nucleus, 2 things will happen. The first thing, the nucleus undergo pycnotic. Pycnotic means condensed. That is the first thing. Pycnotic. And after that, the nucleus undergo fragmentation. Fragmentation of the nucleus is known as karyotaxis. So this is the third change, nuclear change, or you can consider 2 different change. So the first is the cell shrinkage, second is eosinophilia, third is pycnosis, fourth is karyotaxis. Define them, each of them in sequence. The fifth, you can understand the pycnosis is compactness of the nucleus and karyotaxis is fragmentation of the nucleus. Give me a thumbs up. The fifth change, you can see the nucleus is pycnotic. You can see the cell pycnotic nucleus. So pycnosis occurs. You can see electron microscopy. In this cell, this is the pycnotic nucleus. Can you appreciate a pycnotic nucleus? It gets condensed. And in this cell, it is karyotaxis. The nucleus undergo fragmentation. In this cell, it is karyotaxis. So these are electron microscopic diagrams. So the same diagrams, this is fluorescent. Okay. Coming on the fourth or fifth, whatever you say. So the next change is formation of blebs. On the surface of the cells, blebs are formed. So say from this beginning, so this is a normal cell. The first change is cell shrinkage. You can appreciate cell shrinkage here. The second is cell eosinophilia. The second is eosinophilia. The third is the nuclear change, pycnosis and karyotaxis. The fourth one is the blebs. On the cell membrane, blebs are there, but still, mind the word, the cell membrane is intact. Bleb बन गए मतलब ऐसा नहीं defect आ गया. Bleb बने लेकिन cell membrane is intact. You have to write that. Blebs are formed, but still cell membrane is intact. Give me a thumbs up. That is the fourth change. So you can see electron microscopy. This is the normal cell. And this is a apoptotic cell on the surface of which multiple blebs are formed. Appreciate the blebs. Give me a thumbs up. Now, after that, all of them convert into apoptotic bodies. So all the blebs are there and ultimately the cell get converted into multiple small apoptotic bodies. There is no leakage in this process. There is no leakage. There is no leakage in this process. Give me a thumbs up. So this is the next. And in the end, you can see a phagocyte will come and engulf all of them. This is the last change. So can you enumerate the 7 changes? Any one of you, can you help me in enumerating the 7 morphological changes in case of apoptosis? If morphology is coming, you have to draw these and write 1, 2, 3, 4, 5, 6, 7. If mechanism is coming in your exam, you have to write the extrinsic, intrinsic, initiation, execution. Give me a thumbs up. Give me a thumbs up. So who will tell me? The first is the cell shrinkage. The second is eosinophilia. These are shown in this diagram. The third is the pycnosis. The fourth is the karyotaxis. The fifth is the bleb. The 6 is the apoptotic bodies and 7 is the phagocytosis. Can you say them in sequence and show them in the diagram? So that is the 7 morphological features. One by one you have to enumerate. It is cell shrinkage first, eosinophilia followed by it, then pycnosis, then karyoraxis. What was the next? Then blebs formation, apoptotic bodies formation, and in the end phagocytosis. Count, they are 7 in number. 1, 2, 3, 4, 5, 6, 7. Draw them, write them, describe them. That is the morphology of apoptosis. Coming on the diagnosis of apoptosis, there are only 2 things for diagnosing apoptosis. 1, 2. There are 2 diagnoses. What are the 2 ways of diagnosing apoptosis? Can you tell me? I have told you all these changes now, but still you feel, ma'am, I got confused with cell. So it is more pink, it is smaller, but I got confused the rest of the cell and this cell. How I confirm that this cell among all the cell on a slide is apoptotic? So there is a marker. There is a apoptotic marker. The name of the marker is annexin 5. If you write this, I will give you 1 mark in your university exam. Annexin 5. Annexin 5 लिखने के 1 complete mark मिलेंगे, right? So annexin 5 is the marker. In this slide, this cell is smaller than the others. This is more pink than the others. See, the nucleus is pycnotic as compared to others. So based on morphological feature, this is a apoptotic cell. Still, I am confused. Ma'am, this cell is apoptotic or not? I am feeling so, but I am not sure. So you just apply annexin 5 over it. So only apoptotic cell will take the dye. Will take the stain, others will not take the stain. Then wash it. After washing, see the slide again. This cell will convert into brown color, other will remain same. So annexin 5. What is the principle of annexin 5? Why only apoptotic cell among all cell is taking annexin 5 and not others? Because annexin 5 का वही principle है phagocytosis का. Annexin 5 bind with phosphatidylserine. In apoptotic cell, the phosphatidylserine is on the outer surface. So annexin 5 will bind with that. But in normal cell, the phosphatidylserine is on inner surface. So annexin 5 will not stain the normal cell. So among this slide, all the cells have phosphatidylserine on inner surface. But this cell have phosphatidylserine on the outer surface. So annexin 5 will bind with this cell and stain this cell, give it a different color as compared to others. This is the principle. Give me a thumbs up. Same diagram from the Robbins. You can see this is annexin 5. Okay. You can see this is annexin 5 and this is a normal cell. You can see the phosphatidylserine is present on the inner surface. So annexin 5 is not binding here. But you can see this is a apoptotic cell. This is the cell membrane. You can see phosphatidylserine is externalization. And you can see annexin 5 is binding with it and giving it color. Due to the externalization, it is annexin 5 giving the color to the apoptotic cell. The first way of diagnosis. Give me a thumbs up. The second way is electrophoresis. You know what is electrophoresis? In biochemistry, you have already you have already read that what is electrophoresis. In electrophoresis, basically this is a gel electrophoresis. We take the nucleus of the cell, we extract the DNA of the cell and we arrange the multiple pieces of the DNA in a sequence. Okay, let me explain you. So this is a cell, this is the nucleus of the cell. Inside the nucleus of the cell, this is the double-stranded DNA. No, this is a normal cell. It is not an apoptotic cell. If I do the electrophoresis of this cell, I will extract the nucleus, that is the double-stranded DNA, and I will arrange it on a gel. So in the gel, you will find a single big line because the complete DNA is in 1 fragment or 2 fragment, right? So this is a normal cell. Can you see? This is complete 1 line, complete 1 line. This is normal cell. You got my point? But now the cell is apoptotic cell. Now the cell is apoptotic cell. So in apoptotic cell, I told you nucleus में 2 चीजें होती हैं. One is pycnosis. What is the second one? The second one is the karyoraxis. What is karyoraxis? Karyoraxis is fragmentation of nucleus. So the nucleus is fragmented here. It is karyoraxed. The complete nucleus is fragmented. DNA is not in 1 portion, it is multi. Now if I do the electrophoresis of this cell on a gel, so I will get multiple pieces. I will not get a single 1 line like normal. So compare the electrophoresis of these normal cell with a apoptotic cell. In the apoptotic cell, you are getting multiple small DNA because of karyoraxis. Name the enzyme who is causing fragmentation of the nucleus. The name of the enzyme is endonuclease. Endonuclease is the enzyme which causes karyoraxis, the fragmentation of the nucleus, because of which we are getting this pattern. ये कैसा दिख रहा है? हिंदी में सीढ़ी समझता है क्या? Step ladder. It is looking like a step ladder. Step ladder. CD, you know, CD, right? It is looking like a step ladder. This is known as step ladder appearance. So apoptotic cells give step ladder appearance on gel electrophoresis. Give me a thumbs up because of endonuclease enzyme, because of karyoraxis. It is not seen in normal cell. This is how on electrophoresis we can differentiate apoptotic cell from a normal cell. Give me a thumbs up. Come on, I can't see your thumbs up. Rinku, Shweta, Kalma, everyone. Balwant Bhai, everyone. Yes. So it is step ladder pattern which is seen due to the fragmented DNA in apoptotic cell because of the endonuclease enzyme. But normal cell, this is not there. So that is the 2 ways of diagnosing. For diagnosis, I am having 1 marker, that is annexin 5. and next then 5, and I'm having electrophoresis. The gel electrophoresis, I will get step ladder arrangement. If it is a apoptotic cell, I will get step ladder arrangement. Everyone give me a thumbs up. Shall I proceed ahead? Shall I proceed ahead? Yes or no? Yes or no? You want to see again? So this is the diagram. Rinku. So this is the one, right? You want this again now? So in normal, it is continuous, or there are few fragments you can see, but in apoptosis, there are multiple fragments looking like a step ladder arrangement. Give me a thumbs up. Coming on the last point, differences from necrosis. Can you differentiate apoptosis from necrosis? What are the points? How many points are there? Okay. So this is apoptosis, this is necrosis, the two type of cell death. Can you tell me the differences? Apoptosis takes place of single cell. Necrosis takes place of a cluster of cell. Everyone give me a thumbs up. The first difference. In apoptosis, cell shrinkage is there. In necrosis, cell swelling is there. I will teach you necrosis next. We have already seen apoptosis now. The 7 morphological feature, the first was cell shrinkage. Pycnosis, karyoresis is common for both of them. Nuclear changes are common, right? Here, in apoptosis, cell membrane is intact. Cell membrane is intact. It is so there is no leakage and no inflammation. But here cell membrane is disrupted. So there is leakage, there is inflammation. Here leakage, inflammation absent. Here leakage, inflammation present. Here leakage, inflammation absent because cell membrane is intact. Here leakage and inflammation is present because cell membrane is disrupted. This is the biggest change. Now you can see the two things in the two diagrams. See, this is apoptosis and this is necrosis. Let me show you necrosis first. So in necrosis, you can see this is a cell, appreciate cell swelling here. And this is a cell, appreciate cell shrinkage here. You can appreciate shrinkage here and you can appreciate swelling here. If you can appreciate, give me a thumbs up. The diagrams are from Robbins. Give me a thumbs up. Beautiful diagrams. Give me a thumbs up. This is a normal cell. Appreciate swelling and shrinkage. Give me a thumbs up. Have you appreciated? Here, if you appreciate and zoom the diagram, if you can zoom my image, you can see the cell disruption. If you can see, there are gaps in the cell membrane. Because of the disruption, there is leakage. The cell disruption is there, leakage is there, and because of the leakage, neutrophils are coming. That is, inflammation is present. Give me a thumbs up. On the contrary, see this diagram. The complete cell is converting into apoptotic bodies, but if you zoom the image, see there is no cell membrane disruption. The cell membrane is completely intact. There are no disruption, no gaps. Give me a thumbs up. So in the end, a phagocyte is coming and engulfing all the apoptotic bodies. There is no inflammation, no leakage, no inflammation. There is leakage, there is inflammation. Give me a big thumbs up. Everyone give me a thumbs up. Do you have any doubt in that? So that is the thing. We are done with apoptosis. If apoptosis is coming for 5 marks in your exam, I'm expecting you to write everything. Or else, if only mechanism is asked, write mechanism. If only morphology is asked, write morphology. If only differences is asked, write only differences, right? So don't write everything. Your time will be limited in your exam. So you will be able to write only one answer, not others. That you don't have to do, right? So whatever is asked, write that only. And if it is a 5 mark, try to write everything. Everyone give me a thumbs up. Everyone. The most important is mechanism and morphology and the differences. These 3 points are very important. They come separately also. And in the 5 mark, I'm expecting you to write these 3 things judiciously. Rest, okay, types, if you don't write, I don't mind. Diagnosis, if you don't write, I don't mind in 5 mark question, right? So that is the thing. Give me a thumbs up. Everyone give me a thumbs up. If you write, it's good. If you don't write, it's okay. Coming on necrosis, the second type of cell death. So I will be teaching you necrosis in the next 20 minutes. We will finish necrosis also. You want me to continue? Of course, I will continue. So I'm starting the second type of cell death, necrosis. Everyone give me a thumbs up. Necrosis. Necrosis is murder. Apoptosis was suicide. Necrosis is murder. The 2 type of the cell death. So the person who's undergoing murder, he will shout, he will call everyone for help, right? But ultimately he died, right? The people are coming for the help, but they can't help. So ultimately, so inflammation is there. Everyone will come. Neutrophil will come. Monocyte will come. Lymphocyte. There is cell burst. So everything, the leak out, there is inflammation, but ultimately cell will die, right? So that is a thing. Necrosis. How you will define necrosis? So it is a type of cell death in living body. You know, in dead body also cell die, but that is known as autolysis. Autolysis is different from necrosis. Necrosis is cell death in living body, and it is always pathological. It is never physiological. One more difference between apoptosis and necrosis you can add in the table, which I have not written there. In that difference ka table, right? Apoptosis can be physiological as well as pathological, but necrosis is never physiological. It is always pathological. If necrosis is there is some disease. It can never be physiological. Give me a thumbs up. So here, the definition of necrosis is in front of you. It is the localized area of death of a tissue. So the cell dies a group of cell dies, it is not a single cell, it is a group of cell, and it is there the degradation of the cell will burst, the cell will degrade, and enzymes will come out. This is the definition. Here mechanism is not important, here a group of cell is involved, in contrast to apoptosis where it was a single cell. So see the difference. Here inflammation takes place, leakage takes place, in contrast to apoptosis where there is no inflammation, no leakage. See the difference. You got the difference. So here types are important. If apoptosis comes in your exam now, the examiner will ask the mechanism and morphology. Apoptosis के types important नहीं है, physiological, pathological. वहाँ पे mechanism ज़्यादा important है. But if necrosis come in your exam now, the examiner expecting you to write the 5 type of necrosis. Necrosis is of 5 types. What are the 5 types? Triple C double F. The mnemonic is triple C double F. Coagulative. The first one C is coagulative. Second C is collicative. Collicative is also known as liquefactive. Better to say it as liquefactive. But yeah, other name is collicative. The third C is caseous. The third C is the caseous. The 2F, the first F is fat necrosis, then fibrinoid. What are the 5 type of necrosis? It is coagulative, collicative, also known as liquefactive, caseous, fat, and fibrinoid. The 5 type of necrosis. Give me a thumbs up. Come on. So I am expecting you to make this table with me. Again, I am expecting take a notebook, take a pen. Please make this table with me. It will be fun to revise entire necrosis in just 5 minutes just before the exam. You can revise everything from my notes in just 5 minutes. The entire first chapter for teaching I require 2 hours, but you can revise it just in half an hour or just 15 minutes. You can revise everything. Apoptosis, necrosis, cell, the various types of adaptations, the 5 mechanism of the injury, reversible injury, irreversible injury. Once you understood it now, you will never forget. You should have concept, right? Concept is important. It is not the rectification. Pathology is a subject जिसमें learn करने का कुछ भी जरूरत नहीं है. Everything is understandable. It is the pathogenesis of the disease. Everything is so beautifully explained in the form of the diagrams and flowcharts. So nothing to learn, right? Anyways, let me teach you the 5 type of necrosis. The 5 types of necrosis. Thank you, Rinku. Thank you for the feedback. I guess everyone got it, right? Apoptosis mechanism. Now I will teach you the 5 type of necrosis in a beautiful. No one will have explained you the differences between the 5 type of necrosis. No one. I am guaranteed जहाँ पे जितने audience attend कर रहे हैं उनमें से किसी को भी नहीं पता होगा what is the difference between coagulative, liquefactive and caseous necrosis. All of them are the type of cell death. They all are necrosis. They all are necrosis. But what is the difference? You will say, ma'am, if they all are necrosis, they all are cell death. Why 5 different types? So there may be some difference in the 5 now. In the type of cell death also there may be some difference. Yes. So I will explain you the definition of each type, the 5 differences. I will tell you the causes of each of them. I am expecting you to write down the gross of each of them, and I am expecting you to draw microscopy. Microscopy is something not written. Microscopy is something that you have to draw. So draw. Use only 2 pencils, the pink and purple. That is lilac pencil and purple pencil, and you can use red one for drawing it. So draw the diagrams here. Try practicing diagrams if you're a second prof student. If you're preparing for your competitive exam, NEET PG, FMG, INICT, or the next, or any competitive exam, USMLE, so no need to draw. Understanding is much better, right? Okay, let me start the 5 type. Before starting the differences between the 5 type of the necrosis one by one in this table, I would like to explain you something about the cell. In a cell, there are 3 features. What are the 3 features? Can you see the outline of the cell? Outline is the shape of the cell. It is known as architecture. It is known as architecture. If I use the word architecture, I mean the outline and the shape of the cell. Now I will use this word frequently in necrosis. So if I say architecture is there, I mean the outline is there, the shape of the cell. The second is the cytoplasm. There are cytoplasmic details in the cell. What are the cytoplasmic details of the cell? What is the color of the cytoplasm? How much it is moderate, scanty? What is the amount of the cytoplasm? Whether the cytoplasm is granular or non-granular? Is it scanty, moderate, or abundant? What is the color? Is it pink? Is it blue? Right? So that is the cytological details. And there are nuclear details. The third and the last thing is nuclear details. So nuclear details, what is the location of nucleus? Is it central? Is it polar? It is eccentric. What is the size? Is it large? It is small? Is it central? Is it hyperchromatic? What is the color? So these are the details of the nucleus. Looking at the 3 details, the architectural details, the cytoplasmic details, and nuclear details, I can identify the type of the cell. I can give you the identity of the cell. Now in human body, more than 1,000,000 type of cells are present. We have RBC, we have WBC, we have neutrophil, eosinophil, basophil, the muscle cell. See this type of the cells. These all types of cells are present in human body. Kahan gaya? Just a second. What happened? Okay, we have muscle cell, intestinal cell, fat cell, neuron, monocyte, RBC, basophil. We have many cells. So can you identify each of them? Yes, each of them have different shape, different architecture, different cytoplasm ka color, amount, granularity, different nucleus ka color, its content, its location. So based on the three details, we can identify the cell. Give me a thumbs up. For identification of a cell, we require three things: architectural detail, cytoplasmic detail, nuclear detail. Give me a thumbs up. Give me a thumbs up on this point. So these are the basics you must know. Now let me start the first necrosis, coagulative necrosis. Listen, what is coagulative necrosis? In the introduction, I am saying it is a type of cell death in which architecture persists, but cytoplasmic and nuclear details are lost. So cell is dead. So only architecture persists. Inside everything is dead. So cytoplasm details are lost. See the diagram. See the diagram. This diagram I'm talking about. See, this is a group of alive cells. These four cells are live. They have three features. They have shape, they have cytoplasm, they have nucleus. So based on the three details, they are live, right? Now see the group of these four cells. These four cells. So you can see, comparing to this, only outline is maintained. Only outline is maintained. Cytoplasm color is changed. So cytoplasm is non-reliable now. And nucleus is disappearing. Nucleus is ultimately pycnotic, fragmented and disappearing. So cytoplasmic and nuclear details are lost. Here all three details are present. Architecture, cytoplasmic and nuclear. They are present, present, present. Here only architecture is present. So this type of necrosis is coagulative necrosis, in which we have only shapes. We have only borders. We have only outlines. The cytoplasmic details are lost. The nuclear details are lost. Give me a thumbs up. This necrosis is known as coagulative necrosis. So what is the definition of coagulative necrosis? It is a necrosis in which architectural details persist, but nuclear and cytoplasmic details are lost. This is the definition of coagulative necrosis. Give me a thumbs up. Fatafat. It is the most common necrosis among the five. So among these five necrosis which I am going to teach you now, this one is the most common. Here architectural outline persist, but cytoplasmic and nuclear details are lost. Now, have you ever seen a ghost? Ghost का मतलब होता है भूत. I have not seen, but I have heard about it, right? So ghosts have outline. They just have outline. They don't have inner details. They don't have inner details. They just have an outline. The ghost, right? In the same way, the cell also have outline, no inner detail. So these cells are known as ghost cells. Bhoot hai ye. Give me a thumbs up. So in coagulative necrosis, ghost cells are formed. This is the reason why we call them ghost cell. Give me a thumbs up. So we call them ghost cell. This is the reason. The second, we also call them tombstone. You know what is tombstone? What is tombstone? What is tombstone? Tombstone is a stone which is present over a graveyard. So this is a stone. Have you seen a stone? Stone have outline. Stone don't have anything inside. It is blank from inside. So the tombstone appearance. So these cells have two appearance. Either the ghost cell or the tombstone cell. Give me a thumbs up. These are the two names given to the cells undergoing coagulative necrosis. Still, the type can be recognized because the outline is maintained. Give me a thumbs up. So that is the definition. If you got the definition, tell me the causes. You know, human body from head to toe, we are made up of cells, organs. All cell and all organs require blood. What if any organ not getting blood? You will say, ma'am, ischemia will happen. What if the ischemia continues for a longer time? You will say the cell will die. If any cell not getting blood for a more time, the cell will ultimately die. So the cause of the death is coagulative necrosis. So ischemia of any organ of the body, the cell will die. It can be liver, it can be spleen, it can be kidney, it can be intestine, it can be stomach, it can be any, it can be muscle, it can be bone. Any human organ not getting blood, the cell of that organ die because of coagulative necrosis, the first one, except one organ, brain. In ischemia, if ischemia occurring in brain will also die. The cells of the brain will also die, but not by coagulative necrosis. The second necrosis, that is lipified. You got my point? What I mean? So any part, liver, spleen, intestine, stomach, esophagus, bones, muscles, if any of them, and brain on one side, right? If any of the organ not getting blood, and if brain not getting blood, if any of them not getting blood, they all will die because of coagulative necrosis. But if brain not getting blood, it will die because of lipified. So brain is the exception. The exception is the brain. Give me a thumbs up. Everyone give me a thumbs up. So the first example is ischemia. Ischemia. Now grossly, the organ is pale, firm and shrunken and swollen. Can you see a liver? This area is showing coagulative necrosis. It is pale and shrunken. Can you see this diagram? Can you see this diagram? I guess it is a spleen. Can you see this? This portion is necrotic, coagulative necrosis. It is pale, swollen, shrunken. This is kidney showing coagulative necrosis, pale, swollen, shrunken. So gross may learn pale and swollen. That's it. Coming on microscopy. So here, can you see tombstone? The stone is known as tombstone. It is a stone over the graveyard. Cell look like tombstone. Cells will look like tombstone. The cells will look like ghost. These are the two terms we give here. Everyone give me a thumbs up. Everyone. Can you see the two diagrams? Now microscopy is important. This is what students don't know, don't understand what is given in the image. You just see something pink, something blue, something purple is there. But what the author, examiner want you to draw in the exam and we want you to understand is this. Listen now. In this diagram, Can you see the two images? This is normal. This is normal. This side is normal. And this side is coagulative necrosis. Both of them are kidney. Both of them are kidney. Because I can find a glomerulus here. And I can find a glomerulus here also. This is a glomerulus. Both of them are kidneys. And along with the glomerulus, these all are tubules. Along with the glomerulus, these all are the tubules. Give me a thumbs up. Give me a thumbs up. Now compare. Let me draw a few cells here. Okay, I would like to explain you. See, this cell have an outline cytoplasm nucleus. The cell of the tubules have three things: outline, cytoplasm, nucleus. But here I can find only outline. I cannot find anything else. I can find only outline. No cytoplasmic detail, no nuclear details. Cytoplasm color is changed. It become light pink in color. And the nucleus are lost. It become pycnotic. So only outlines are maintained, but inner details are lost. That's why it is known as ghost appearance, tombstone appearance, and it is a diagram of coagulative necrosis. I can still see the cell. Cell is there. These are the tubules. I can still see the cell, but inner details are lost. I can see the outlines. See, I'm drawing the outlines. So outlines are maintained. I can compare them. Okay, this is this, this is this, this is this. It is corresponding. Outlines are maintained, but inner details जो इसमें है वो पूरी यहाँ पे गायब हो गई. So cytoplasmic and nuclear details are lost. Give me a thumbs up. So the cells are tombstone or ghost appearance. Give me a thumbs up. At the junction, you can find lymphocytes. That is inflammation. Can you see all these cells? What are all these cells? These all are lymphocytes. So that is inflammation. So inflammation is the hallmark of necrosis. Necrosis cannot occur without inflammation. At the junction, there is always inflammation. So that is the diagram of coagulative necrosis. Give me a thumbs up. Don't write the details. Just understand. Tombstone appearance is there. I have shown what is tombstone appearance. That is outlines are maintained, but nuclear and cytoplasmic details are lost. That is tombstone appearance. And there is inflammation at the junction. That's it. This is there. Now, not only kidney, in any body part, this diagram can be drawn, except brain. Brain mein nahi hota coagulative necrosis. Except in brain, you can draw the diagram of any part. Now, I have drawn two diagrams to show you the comparison. This is normal, this is coagulative. Give me a thumbs up. Give me a thumbs up. So, tombstone appearance are there. That is the only information given in this diagram. After that, I would like to come on the next liquefactive necrosis. Can you help me in filling coagulative necrosis? In the introduction, write down the definition. Architecture is maintained, but cytoplasmic and nuclear details are lost. That's why the cells are known as ghost cell. That's why the cells are known as tombstone cells. So that is the introduction. And it is the most common among the five in the introduction. In the causes, write down only one cause: ischemia. Ischemia of all organ except brain. Don't forget brain. Except brain. Brain yahan pe aayega. Ischemia of brain is an example of liquefactive necrosis. Gross may write down the organ is yellow, pale, swollen. Microscopy may draw the diagram. On one side show normal, on other side show coagulative organ, kidney, it can be kidney, but you show the only outlines are maintained. Rest of the details are lost, and at the junction show the inflammatory infiltrate. So that is the coagulative necrosis. Coming on liquefactive necrosis. Liquefactive necrosis or it is also known as colligative necrosis. Just a second. Liquefactive necrosis. What is the definition here? In liquefactive necrosis, all three details are lost. In liquefactive, all three. So architecture is also lost. Cytoplasmic is also lost. Nuclear is also lost. So you say, ma'am, what will remain? Nothing will remain. A liquid will form. Can you see a cluster of live cell having all three details? There is architecture, there is cytoplasm, there is nucleus. I can see. But after liquefactive necrosis, everything disappeared. So I can't see the outline. Architecture is lost. I can't see the cytoplasm. It is lost. I can't see the nuclear, it is lost. So what is formed? A liquefied material is formed. A semi-liquid material, a semi-liquid jelly-like material is formed. That's why known as liquefactive necrosis. Give me a thumbs up. Now see the difference between the two in coagulative and liquefactive. You want to compare? Do a comparison. In coagulative necrosis, the cell, here see the coagulative necrosis. In liquefactive necrosis, see the liquefactive necrosis. So what is the difference? Who will tell me the difference? Here architecture is maintained, but cytoplasmic and nuclear details are lost. Here all three details are lost. Architecture, cytoplasmic and nuclear details all are lost. Here since architecture is maintained, so it is ghost cell or tombstone cell. Here all three details are lost, and ultimately a liquid is formed. A semi-liquid or jelly-like material is formed. Give me a thumbs up. See the details now. The diagrams are in front of you. So this is the definition. After the definition, come on the causes. Causes is ischemia of brain, which was an exception there. So it is included here. Number one. And number, biogenic bacterial infection. So you know bacteria occurs can come in any part of body and cause the infection. So bacterial infection of any part of human body can cause the death of that body part or cell by liquefactive necrosis, right? Bacteria form pus. Pus is liquid. So pus is actually the cell, all the things got converted into liquefied material, the architecture, the cytoplasm and the nucleus. Give me a thumbs up. So these are the two examples. Gross में a liquefied center containing a cyst wall will be there containing a liquefied center. See, this is brain. It occurs in brain now. So this is the diagram of the brain. In the brain, can you appreciate a cyst? This is a cyst wall. Inside it, appreciate the liquid. Appreciate the liquefied cavity. So this is the gross. Coming on the microscopy, this is the diagram. So I will show you diagram of brain here because it occurs in brain, right? Coagulative, I have shown you the diagram of the kidney. Here I have shown you the diagram of the brain. Now in this diagram, what the author wants to tell us, very, very beautiful diagram. I like this diagram. Now see, this is the junction. Again, this is the junction. On one side, this is normal brain. And on other side, this is liquefactive necrosis. This is liquefactive necrosis. On this side, this is liquefactive necrosis. And this one is normal. This one is normal. Give me a thumbs up. Now, normal brain, can you see the cell? Yes, I can see these are the neurons. These are the oligodendrocytes. These are the neurons and oligodendrocytes. The background is the fibrillary. This is a normal brain. I can see the cell. I can see the outline of the neurons. I can see the nucleus. I can see the cytoplasm. All three details are visible to me. Neurons, oligodendrocyte, various cells are there. Background is fibrillary. It's very beautiful. It's normal brain, right? See on the other way around. See this side. What you can see. See in this portion. See in this portion. I can't see anything. I can't see anything. What I can see in this portion is dot, 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 dot. Not even single cell. Not cytoplasm, not outline, not cytoplasm, not nucleus, no, not outline. I can find a liquid. This is liquefied debris. This is liquefied debris I can find. And at the junction of the two, I can find this one. Can you see the junction? In junction, you can find this is the junction. In the junction, you can find the inflammatory cells, lymphocytes, neutrophils with the blood capillaries. So this is known as granulation tissue. So at the junction, there is granulation tissue which will form the cyst wall. So on one side, normal brain in which all three findings are there: architecture, cytoplasm and nucleus of the brain. On the other side, see the liquid debris. See, appreciate only dot, 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 dot. This is all liquefied debris. And at the junction, appreciate the granulation tissue. Give me a thumbs up. Give me a thumbs up. That is lipified necrosis. Will you be able to draw these findings in your exam? I guess you can. So here, draw the two kidneys, normal and coagulative. Here, draw the two brains, normal and liquefactive. And at the junction, here shows only inflammation. Here, along with inflammation, show capillaries also. That is granulation tissue. That is the only difference. Give me a thumbs up. Coming on the third, Caseious necrosis. Who will tell me Caseious necrosis? What do you mean by Caseious? The word Caseious matlab cheese. I like cheese. Do you eat cheese? I guess yes. Everyone likes cheese. In pizza, everywhere, we like cheese. Yes or no? The word Caseious means cheese. Why here necrosis is known as cheese necrosis? Why? It looks like cheese. Because how is cheese? Have you ever seen cheese? It looks like that. It is dry. It is granular. It is dry and granular. It is dry, granular and yellowish in color. Yellowish in color. So necrosis will also look like that. That's why it is known as Caseious necrosis. Okay, ye to hua appearance. What is the definition? Here also all three details are lost. So can you see a group of live cells here? Yes, you can see. Here, the architecture is lost, cytoplasm is lost, nuclear details are lost. So you will say, ma'am, then what is the difference between liquefactive and Caseious? Wahan pe bhi to तीनों details lost ho rahi thi. In liquefactive, all three details were losing and cell was converting into a liquefied debris. Here, all three details are lost, but cell do not convert into liquefied debris. Cell convert into a dry granular debris. Here, granular debris are formed. So that is the only difference. Give me a thumbs up. You can see the three type of necrosis in front of you. Here, architecture is maintained, but cytoplasm and nuclear are lost. That's why cell look like a ghost or tombstone. Here, all three details are lost. Cytoplasmic, architecture, nucleus. Here also all three details are lost. Cytoplasmic, architecture, nucleus. But here liquid debris are formed and here granular debris are formed. I cannot simplify more than this. Please appreciate the three definitions. So if someone asks you the definition of coagulative necrosis, this is the definition. Architecture persists but nuclear cytoplasmic details are lost, so ghost cell or tombstone cells are formed. What is the definition of liquefactive necrosis? All three details are lost and ultimately cells convert into a liquefied debris. What is the definition of Caseious necrosis? All three details are lost and cell converted into a granular debris. Give me a thumbs up. Draw these diagrams and write the definitions in your exam. You will get full marks. Everyone give me a thumbs up. You got my point? So that is the definition. And the organ look like a cottage cheese because it is dry, granular, yellowish. So architecture and cytoplasm and nuclear, all details are lost and cell converted into a granular amorphous debris. It is not liquefied. So that is the definition. So causes, 4 causes are there. यहाँ पे 4 causes हैं. 2 bacteria, 2 fungus. So Mycobacterium Tb, the first bacteria, syphilis, Trypanosoma pallidum, the second bacteria. These are the two bacteria. Two fungus are Histoplasma and Coccidio. So these are the 4 examples here. Here appearance is dry cheese-like. Can you see this is the organ? Can you see this is the lung? Inside lung, this is the TB. Can you see that this is looking like cheese? This one is looking like cheese. Yes, dry, granular and yellowish white in appearance. So that is the thing. Microscopically, this is a lymph node. Can you see a lymph node? This is the cell. This is the capsule of the lymph node. Just below that, normal lymph node is there. Now, right, in normal lymph node, we have follicles. Can you appreciate the follicles? We have follicles. In the follicles, we have lymphocytes. The lymphocytes, I can see the cytoplasm, I can see the nucleus, I can see the outline. So these are the follicles inside which lymphocyte ka I can see 3 things. So this is a normal lymph node. On this side, this is normal. I can find architecture, cytoplasm, nucleus of the lymphocyte, normal lymph node. See this side, what you can see? Only pink color mass. What you can see? Can you see a side as architecture? No. Can you see cytoplasm? No. Can you see nucleus? No. What you can see? Pink color homogeneous material. So entire cells, these cells, they got necrotic and the granular debris are formed. Homogeneous granular cheese-like debris are formed. And at the junction of the two, I can appreciate the inflammation. This is the inflammation. But this inflammation is a special inflammation. It is known as granulomatous inflammation. Granulomas are formed. So you can appreciate the giant cells and epithelioid cells. So these are the giant and epithelioid cells there. So that is the diagram. Give me a thumbs up. On one side, normal lymph node. On other side, necrotic lymph node. This is Caseious necrosis. At the junction, you show inflammation, but a granulomatous inflammation. That is the diagram. Give me a thumbs up. So here I have drawn 2 kidney. One is normal. One is coagulative necrosis. At the junction, normal lymphocytes are there, normal inflammation. Here I draw 2 brain. One is normal, one is necrotic. And at the junction, I draw granulation tissue, right? That is lymphocytes along with blood capillaries. Here I draw two lymph nodes. One is normal, one is necrotic. And at the junction, I draw granuloma. That is granulomatous inflammation. Here I have shown the cells only outline and no cytoplasm, no details. Here I draw dot liquid and here draw pink color homogeneous material. That is granular debris. So this is, and on one side you have to draw the normal one. So this is the best way I can explain you the morphology here. So this is how you have to draw the diagrams in your exam, right? Coming on the 4th one, fat and fibrinoid, the 4th and the 5th. These are the 2 special type of necrosis. Fat necrosis occurs in the body where is excessive fat. Human body, 3 places excessive fat. What are the 3 locations? Female breast. Female breast have excessive fat in that, not male breast. Number 2, this is female breast, number 1. Number 2, pancreas. Human pancreas have excessive fat. And number 3, mesentery. Mesentery of the intestine have excessive fat. So death occurs of these cells. These cells contain fat cells, that is adipocytes. These cells contain excess of adipocytes inside them. Give me a thumbs up. So if death of adipocyte occur inside these organs, it is known as fat necrosis. So that is the definition of fat necrosis. So fat necrosis is the 4th necrosis, in which what happens? In the fat necrosis, the fat rich location, the adipocytes are dead. The adipocytes are dead. So it is the breast, female breast, it is the pancreas. 3rd, mesentery is not written, mesentery omentum, you write that also. So these are the 3 causes. Give me a thumbs up. Grossly, it will be chalkish white or yellowish white. Can you see? This is the breast, female breast. You can see this one, this one, this one. So this is all fat necrosis. In the pancreas, you can see this one. It is chalky white. Chalky white in appearance. The word is chalky white. Gross is not very important. If you want to learn, it's good. Microscopically, this is the diagram. Can you see? I don't know the organ because in this diagram, the normal is not shown. Only the necrotic one is shown. This is all necrotic. This is all necrotic. The death of adipocytes. The death of adipocytes. It can be breast, it can be pancreas, it can be, I guess it is breast. So most of adipocytes, they are dead. The appearance is cloudy. The typical word is cloudy. Badal ke jaisa dikh raha hai. So try to draw the multiple clouds like structure in your exam. So the appearance is cloudy and you have to write the appearance is typically cloudy appearance. Typically, there is the word here to be picked is cloudy. And what else? ये बचा हुआ पूरा क्या है? What is this all? This all is inflammation. It is mixed inflammation. It is neutrophil also and lymphocyte also. It is a mixed acute and chronic inflammation. I'm done with fat necrosis. The last one is the fibrinoid necrosis. Fibrinoid necrosis, now it occurs in blood vessel. This is a special necrosis which occurs in blood vessel wall. Blood vessel wall have 3 layers: intima, media, externa. It occurs in media. So can you see? It occurs in the wall of the blood vessel, due to vasculitis. The reason is the vasculitis. Can you see? This is a wall of a blood vessel. Can you see? This is the lumen containing blood. This is blood in the lumen. Can you see? Now this is intima. Intima is normal. There is no problem in the intima. See media. In the media, can you see a pink color ribbon? Ribbon-like material is deposited in the media. Can you appreciate this pink color ribbon here? This pink color ribbon is fibrinoid necrosis which occurs in media. That's it. So I am done. So can you help me in filling this table? Can any one of you, Shweta, Pratyaksha, Rinku, Chali, Kalma, anyone help me in filling this table? So we are done with the 5 types of necrosis. Please help me in filling it. First, tell me the definition of all 5. Can you tell me the definition of coagulative? Yes, architecture is maintained. Architecture is maintained, but cytoplasmic and nuclear details are lost. So cells are ghost or tombstone. Here architecture, cytoplasm, nucleus, everything is lost and cells converted into liquefied debris. Here also architecture, cytoplasm, nucleus, everything is lost and cell convert into granular debris. Here it occurs in fat-rich location, that is female breast, that is pancreas, and that is mesentery or omentum. Here it occurs in the wall of the blood vessel in the media. That is the definition. Everyone appreciate the definition. Give me a thumbs up. If you got the definition, give me a thumbs up. Mujhe thumbs up do if you got the definition. Yes, after definition, I would like to write the causes. Can anyone of you help me for the causes? You must help me for the causes. What are the causes? So here cause is ischemia. Ischemia of all organs except brain. Here ischemia of brain. Here ischemia of brain along with pyogenic infection, pyogenic bacterial infection. Here 4 causes are there. 2 bacteria, 2 fungus. The 2 bacteria are Mycobacterium tuberculosis and syphilis. The 2 fungus are coccidioidomycosis and histoplasma. Here the 3 organs, that is breast, pancreas or mesentery ka either trauma or infection inflammation. Either trauma or infection inflammation. Here vasculitis. The cause is the vasculitis. Everyone give me a thumbs up. Appreciate the causes. Everyone. Shall I come on the gross? Gross is not very important, but yeah, you must know. What is the gross? Here the gross is pale, yellow, swollen, right? Here the gross is liquid, cyst. The cyst containing a liquid. Here the gross is like cheese, dry, granular. Here gross is chalky white, and here no gross is there, right? Coming on the best part, that is microscopy. Who will tell me? Here you have to draw 2 diagrams of the kidney, right? At one side normal, at one side you have to draw only outline, no cellular and nuclear detail. So tombstone or ghost appearance. Here you have to draw diagram of 2 brain. On one side normal, on other side you have to draw the liquid debris. Here you have to draw the diagram of 2 lymph nodes. On one side normal, on other side pink color homogeneous dry granular material. Here you have to draw the diagram of 2 breast, normal breast, and here the cloudy appearance. Here it will be cloudy. And in the last you have to draw the diagram of a blood vessel, only one blood vessel. Intima, media, externa. In the media show a pink color ribbon. So I'm trying to draw the 5 diagrams I have already shown you. So this is the diagram of coagulative necrosis, the 2 kidneys. This is the diagram of 2 brains, liquefied necrosis. This is the diagram of 2 lymph nodes, normal and necrotic, that is Caseious necrosis. This is the diagram of fat necrosis. Appreciate the cloudy appearance here. And this is the diagram of blood vessel showing fibrinoid necrosis. Appreciate the ribbon here, the pink color ribbon like material. I am done with necrosis. Everyone give me a thumbs up. Everyone, jitne audience utni thumbs up. Please appreciate my efforts. I have done complete apoptosis. I have done complete necrosis. In apoptosis, the mechanism, in necrosis, the type of the necrosis. I am done with the first chapter. In the first chapter, all the important points covered by me. So I have taught you the cell depletion, cell injury, apoptosis, necrosis, cell death, mechanism of cell death, and the various types of the cell injury, the mechanisms of the cell injury. Everyone give me a thumbs up. Everyone. So was this lecture useful for you? If you like the lecture, before leaving the lecture, don't forget to click on the like button and don't forget to drop a comment in the comment box. And please share the link of this lecture on your batch groups with your friends, colleagues, seniors, juniors, all second prof students and any student preparing for any competitive exam. Because these topics are not only important for the university exam but also for the competitive exam, NEET PG, FMG and INICT. I would like to take a break for 15 minutes and after that I will continue my lecture. So after taking a break of 15 minutes, I will start the lecture at 12 p.m. sharp. It's 11:45 now, right? So at 12 p.m. sharp, I will start the lecture. It is a free lecture. Where? But not on YouTube. Now I'm continuing the lecture on Unacademy app. So Unacademy Learners app. You have to come on Unacademy Learners app. There is a free lecture, that is episode number 3. We have completed episode 1, 2, now episode number 3 and 4. The next 2 episodes on Unacademy Learners app. It is free like YouTube only, and you will have a better experience there. So please, those who are the newcomers, install Unacademy Learners app from the Play Store. Go to NEET PG category. Select the goal as NEET PG. It is a category for all medicos. Whether you are preparing for NEXT or USMLE or whatever you are preparing, select the category as NEET PG if you are a medico. And after that, search my name. My name is Dr. Priyanka Sachdev. You will find my name in Educators, and once you click my profile link there, you will find a list of recordings of all the free lectures till now. You can get the PDF of the notes also. So I request all the newcomers to come on the app, please. And right now you will find me live after 15 minutes at 12 p.m. there, so you can attend my live lecture also. The only thing, the newcomers will require a code to join my lecture. The lecture is free, but if you are a newcomer, the code will be asked. The code is my surname only. The code is Sachdev. SACHDEV. Sachdev without any space, it's 10. Sachdev 10 is the code. Please use this code for unlocking any free class on Unacademy, not only of me, of any educator you can unlock using the code. So help me in distributing this code to all the medicos throughout the globe. So wherever you have any medico friend, your batchmates, your colleagues, seniors, juniors, interns, everyone, please distribute the code so that everyone can get benefit from the free education which is there on the Unacademy. With this code, you can unlock any free recording, not the paid one. For the paid one also, you can purchase the plans. The plus iconic plans are available, and using the same code, you can get discount. You can get 10% discount on the total price if you apply this code before payment. So code is same for free to unlock and for paid one to get the discount. Thank you very much. I would like to see all of you at 12 sharp. Bye-bye on the app. Bye-bye.